Good morning, everybody. I'm Dr. Jaswinder Singh. I'm assistant professor at the Department of Molecular Cellular Medicine. And the topic that is given to me is bile microbiome and CAGB. First of all, I'm not an expert in CAGB, nor microbiome is my area, but we have developed an area which is called as Meta-proteomics, which is complementary to microbiome. So, to start with, CAGB is the fifth most common gastrointestinal malignancy, and if you see the incidence, it is 9 per 10 million in India. So, if you see the tumor progression, the tumor develops into the gallbladder, it invades the duct and blocks the bile flow. The bile backs up into the liver, therefore, you have different complications of the liver, where you can see the jaundice happening, you have itchings and other things. But what are the risk factors? Cholestasis. In 75 to 98% of the patients with CAGB, cholesterol, different type of stone, old age, being female, typhoid carriers, and other chronic inflammations and IBDs could be one of the risk factors. The relative risk is the amount of the diameter of the stone. The bigger the stone, the higher the risk of development of CAGB. That is known. So why bile in CAGB? So bile is stored in the gallbladder. Everybody knows. So if there is a development of carcinoma in the gallbladder, the bile may recapitulate all the processes that are taking place. Therefore, analysis of bile in gallbladder carcinoma is very key. So what is metagenome? Just to give you a small idea, metagenome is identification of bacteria, different type of phylums, their phylum, class, order, by analyzing the DNA, bacterial DNA. But what we do in our laboratory is we analyze the bacterial proteins and we map it into the different database to identify different bacterial phylums, their classes and other things. The results are similar to what we find in metagenome, but the metaproteome gives us one good advantage is that we could also identify the enzymes which gives us the functionalities of the bacteria. So by doing metaproteome analysis, you may be able to identify which bacteria is actually doing what kind of functions. So there are many publications in the area of gallbladder carcinoma and microbiome. For example, this is a publication in in 2019 where they have screened 3 LD individual bile sample and they have done the metagenome analysis and did the COGS function. These are the COGS function of the bacteria that they identified. And what they said that in the biliary microsystem, the main bacterial phylum were represented by Firmicutes, Bacteroidetes, Actinobacteria and Protozoa. And in patients with cholestasis, members of the family Bacteroidetes and Pervitola and Venolocaceae, these were frequently detected. Another paper which was published in 2020, metagenomic study, they compared gallbladder carcinoma with chronic calcareous cholecystitis, and they took 7 patients. These are the demographic profile of the 7 patients. By doing the metagenome analysis, they could see that the gallbladder carcinoma and triple C was significantly different. The alpha diversity documented by the Shannon and Simpson was significantly higher in the gallbladder patients, and these are the bacteria which they found by IDS scores which are specific to gallbladder carcinoma or CCC. So this study has presented the first evidence that the existence of an altered biliary microbiota in GBC which is clearly different to that of the CCC patients. There are other papers which was published in BMC microbiology. Here in this paper what they have done is they have taken 244 patients who were diagnosed with cancer, cystic lesion and inflammations, and they found a significant difference in the relative abundance of Citrobacteria, Enterobacteria and others. These findings suggest that they have pancreobiliary tract cancer patients have an altered microbiome composition which might be biomarkers. So what they have done is they have done the microbiota. This is the bile duct, this is the pancreas and gallbladder. And on analysis, what they've done, they made 2 clusters based on the microbiome, and the clusters were subjected to Kaplan-Meier. The cluster 2 was showing lower survival in the bile duct lesion, and this cluster 2 was enriched in Enterococcus. So this they have done. There's another paper which compared the bile to ascites, to blood and the stool, and they did the bacterial identification in the bile, in the ascites, in the blood, in the stool, in PSC patients, and then they compared it to the control. What they said that when PSC is compared to cholestasis or cholangitis, so these are the bacteria which was significantly different, and they found in summary confirmed that the published data on the bile microbiome composition in PSC patient emphasizes the reproducibility of bile microbiome, and they show that the composition of microbiome differ between patients exhibiting different biliary pathologies. So with different biliary pathologies, the bacteria that are accumulated in the biles are different. But what we have done at ILBS is, in collaboration with the Department of Surgery, we published this paper in this year 2022, that bile multiomic analysis classifies lipid species and microbial peptide predictive of carcinoma of gallbladder. So in this paper, what we have done is, we have taken a training cohort of 87 patients and a test cohort of 38 patients, where we have the bile samples and paired plasma samples. We did the lipidome isolation. This is the demographic profile of the patients. This is the training cohort and this is the test cohort. If you see, the liver parameters decreased in the training as well as the test cohort. The bile acids and the lipid levels were significantly downregulated both in the training and the test cohort in the CAGB patients. So this is the statistical flow chart. What we have done is, when we did the lipidome analysis, the lipidome analysis was subjected to different statistical analysis. For example, PLSDA, OROC analysis to identify candidate biomarker. The candidate biomarkers were then validated in two different set of cohorts, the cohort 1 and cohort 2, that is bile and plasma, and we also validated the machine by using machine learning approach. The meta-proteome analysis was done and it was correlated to the lipid to identify what bacteria actually correlate to which type of lipid. So what bacteria actually are regulating those kind of lipids. So we did the correlation network analysis and the pathway analysis. To go forward to the composition of bile, this is Macquarie's triangle, which is the percent cholesterol, percent lecithin and bile salt. It gives us the composition of bile. So when we did the analysis using this, what we found that the metastability of bile was significantly lower in patients with CAGB. So you can see all the CAGB patients are accumulated towards the higher side of the percent cholesterol as compared to the others. When we did lipidomic analysis, when we compared CAGB with gallstone or CAGB with healthy control, we found 400 differentially expressed lipids when CAGB was compared to gallstone, and 419 differentially expressed lipids when CAGB is compared to healthy control. Based on this lipidomic profile, you can clearly see that the CAGB was significantly different from gallstone and healthy control. The lipid species were then classified into the classes, and the classes expression was given. You can see the lipid classes like phosphatidyldienestol, phosphatidylethanoamine, all these classes were significantly downregulated in CAGB and they directly correlated to each other. What we did is that, based on the lipid profile, species, we did a random forest analysis and identified top 5 lipid species which could be used for analysis or probability of cancer development analysis. So we did the probability analysis and we could find the probability was greater than 95% with an AUROC of 1. Then we analyzed, as Dr. Gayatri also said, it is very critical to analyze CAGB with and without gallstone. We analyzed our patients with CAGB with gallstone and without gallstone. The lipidomic profile showed 24 differentially expressed lipid, and the BCA profile was significantly different, and the lipid which was significantly higher in patients with gallstone is phosphoinositol. We also did the probability calculations and based on the top 5 differentially expressed lipid, and we found that the probability was around 85% with an AUROC of 84.3. Then we did the bile meta-proteome analysis in CAGB patients. So by doing the bile meta-proteome analysis, we were able to identify 17 phylums. This is patients with CAGB with gallstone, this is with gallstone, this is only gallstone healthy control, and we were able to identify more than 50 phylums. The alpha diversity based on the Shannon and Simpson index was significantly high in the patients with CAGB as compared to gallstone or healthy control, and the beta diversity was also different. We did the functional analysis based on the enzyme that we can identify, and we found that these are the CAGB with gallstone. We found that the bacteria in CAGB gallstone was significantly increased in lipid transport and metabolism. They were also doing different functions such as cell cycles and carbohydrate metabolism. Now what we have done is, again we did the same thing, identified 5 different bacterial peptides and did the probability calculations, which is called as the POD, probability of disease, and what we found that the probability here was greater than 85% with an AUROC of 1. Again, we did the meta-proteome analysis in CAGB with and without gallstone. On comparing with and without gallstone, based on the bacterial profile, we can see that both the groups were significantly different, and the bacteria which was significantly higher in patient with gallstones were lactobacillus, and mycobacteriaceae were significantly higher. So we did the functional analysis and we found that the amino acid transport and metabolism, cell wall membrane and envelope development, intracellular trafficking and vascular development was significantly high in CAGB, whereas signal transduction mechanism, replication and transribosomal structure and biogenesis was significantly high in patients with gallstone. We again identified 5 different bacterial species which were significantly identifying patients with gallstone, and did the POD development. We found that the POD AURC was greater than 90. Then we did the meta-proteome analysis of patient with non-cholestasis versus cholestasis in CAGB patient. You can see that the alpha diversity was significantly high in non-cholestasis patient, and the beta diversity was not that different. But we could identify different bacteria. For example, the mycoplasma, the Normocordia and the Bacillus were significantly higher in the cholestasis, whereas the Actinomycetes and Streptomyces were significantly higher in the non-cholestasis patients. Then we wanted to integrate the lipidomics and the meta-proteomics area results. To integrate, what we have done is a weighted lipid correlation network analysis and the weighted meta-proteome correlation network analysis. Now what it is, it performs an unsupervised hierarchical clustering of the most dysregulated molecule and group them into modules. So what we have done is we have made lipid modules and metaprotein modules, and then identified those modules which were disease specific, for example CAGB specific, and we made a multi-modular correlation network. We cross-correlated these modules to identify this multi-modular correlation network. So this is the result. So this is the result of the WLCNA. So you can clearly see by doing WLCNA, we were able to identify this. This is a single lipid module which was specific to CAGB, and by doing meta-proteome WMCNA, we identified modules of bacteria for CAGB, modules of bacteria for gallstone, and modules for bacteria for healthy control. Now it is very important to identify which module of bacteria correlates with which modules of lipid, so that we can do the association analysis. And these modules carries the lipids which are from the same pathway and bacteria which are doing the similar function. So when we correlated, what we found was the MP blue, MP brown and MP grey, which were associated to gallstone or healthy control. They correlated with directly correlated with all the lipids. You must remember the lipids were significantly downregulated in CAGB, and these metaproteins were correlating directly with the lipid. Only one metaproteome module, which is called as MP turquoise, inversely correlated with all these lipids. So on correlation, again, what we find, the bacterial, we devised the pathways, and these pathways were back correlated to the lipid module, and we found that the bacterial pathway specific to CAGB correlated inverse with all the lipid molecules, directly with all the lipid molecule, and inversely with the black module. That therefore, what we could get from this analysis. So what, by this analysis we devised, the gist of this analysis is, we identify certain bacteria which were known in cancer, we identify certain bacteria which was unknown in cancer, but we found them to be associated. They were performing different types of bacterial functions which were significantly increased, and it directly correlated with different lipid species. So these bacteria are now regulating these lipids by doing these functions that we found. And for example, proteobacteria is downregulated in CAGB. It downregulates the functions of nucleotide transport and metabolism, and it inversely correlated with this bacteria. Again, we correlated the metaproteome modules and the lipid modules with the clinical features. If you can see, this is the turquoise cluster, and the red lines are directly correlation, and the blue lines are inversely correlated. The turquoise cluster was significantly increased in CAGB. As it was significantly increased, it would directly correlate with all the bile acids which were there, and all the clinical parameters which is there. For example, total bile acid, all these bile acid SAP, and the clinical factor, for example, AST, ALT and others. and they inversely correlated with the serum albumin and all the lipid parameters. So this shows that we found a meta-protein module which directly associates with the lipid module as well as the clinics, and it follows the same trend as it goes along. These meta-protein modules, the top 5 bacterial lipid species which we identified are now listed here. When cancer was compared to non-cancer, this is the best lipids. When CAGB was compared with gallstone was compared to CAGB, these are the best lipid species. When gallstone was compared to healthy, these are the best lipids. And when meta-proteins, when cancer was compared to no cancer, these are the bacteria, best bacteria and their peptides. When CAGB was compared to CAGB, gallstone was compared to CAGB only, these are the peptides, and gallstone was compared to healthy control. So we decided we could validate any of them. We decided to validate these 8 set of lipid species for development of cancer. So in validation, what we did the validation and we could identify these are the lipid species. This is at the training cohort, this is the AUC at the training cohort, the p-value, the fold chain, the bile validation, the AUC at the bile validation, the p-value, the fold chain, the plasma validation, the AUC at the plasma validation, the AUSC of fold chain. You can see that all these lipid species were validated in both of them, and this is the LCMS validation for the lipid species which we have. Then we did the machine learning analysis, the accuracy all together, all of the 8 together, and individually we did the accuracy determination by using machine learning for 4 different models. For example, we did the LDA model, KNN, SVM and random forest, and we found that both the accuracy as well as the kappa value was significantly higher in all together as compared to the individual. We did the accuracy and accuracy determination in both the primary, the first test cohort and the second test cohort, that is the bile cohort as well as the plasma cohort, and you can see the accuracy, the sensitivity, the specificity and the p-value was significantly higher on all together in both the cohort, in the plasma cohort as well as in the bile cohort. So by this what we can say is we found first the global suppression of lipid in bile is a characteristic feature of patient with CAGB and it correlates to severity. Second, CAGB patient have a distinct and a unique lipid fingerprint composed of lipid group, phosphatidylcholine, phosphatidylethylamine and others. We found increase in phosphatidylethylamine isn't specific for gallstone development. We were able to identify CAGB gallstone in healthy bile associated microbiome signature and their functionality. The microbiome diversity in CAGB patient as compared to gallstone and healthy were higher and could be a probable reason for development of cancer in CAGB. The integration analysis describes CAGB associated lipid modules and microbiome modules. Multimodular correlation analysis suggests the contribution of bacterial microbiome in lipid module expression. We show that the CAGB associated modules are directly associated with liver functions and bile acid profile, including primary and secondary bile acid. And finally, comparing the 4 machine learning algorithms for the 8 panels of lipid species, we provide evidence that the identified lipid panel correlate with collectively serves as the most reliable indicator, and random forest model serves as the most preferred ML algorithm for CAGB detection in bile as well as paired plasma samples. Now, to way forward, we are now analyzing certain pediatric cirrhosis work in collaboration with the pediatric department, and this is in 161 patients. I just wanted to show you certain glimpse of the cirrhosis versus non-cirrhosis that we identified. This is the plasma microbiome or meta-proteome analysis, and you can see based on the plasma meta-proteome analysis, cirrhosis has a higher alpha diversity as well as the beta diversity as compared to non-cirrhosis in pediatrics. And these are the bacteria which are, sorry for the names are very small, but we identified bacteria which are cirrhosis specific and non-cirrhosis specific in pediatric cirrhosis. And when sepsis was compared to non-sepsis in the same patient, the cirrhotic group, we found that the sepsis had a higher alpha diversity and the beta diversity was this, and the bacteria which could identify which is non-specific or sepsis specific, were identified, streptococcus and other bacteria were identified to be specific. With this, I would like to thank my mentor, Professor Shiv Kumar Sarin, and my team, and I'm on for questions. Thank you.